Hello, I'd like to welcome you all to the fourth in our series of the 11th annual Science Lecture Series here at UC Riverside. I'm Francie Sladek, the Divisional Dean of Life Science and Professor of Cell Biology and Toxicologist. Before we get started, I'd like to ask how many of you have been to any of the previous three seminars in this series? Okay, good. Lots of people. Okay, good. How many of you have been to more than one? Okay, good. How many of you have been to all three so far? Okay, good, great. Well, so we got a lot of permanent fans here. That's wonderful. I'd also like to thank the science ambassadors. They're the young people in the blue t-shirts. They're some of our top students in the College of Natural and Agricultural Sciences, and they help with events throughout the year. I would also like to thank teachers, particularly those from some of the high schools that really encourage their students to come. Do we have anyone here from North High School? Yeah? Yeah. How about Murrieta Valley? Yeah? Santiago High School? Yeah, I see one or two. Okay. University Heights Middle School? Can't see it. Okay. Ramona High? University Prep High School? Temecula Valley? And what about the community colleges? Do we have anyone here from Riverside Community College? Okay, good. Norco. All right. Moreno Valley and San Bernardino Valley. Okay. Well, I'd like to welcome you all. I'd also now like to ask, I know I see a lot of current UCR folks in the audience. How many UCR alums do we have in the audience? Okay, good. All right. Thank you. Well, welcome back. All right. So I'd also like to thank some of our co-sponsors for this event. This is mainly put on by the College of Natural and Agricultural Sciences, but 91.9 KBCR also sponsors this event, as well as CAFE, the California Agricultural and Food Enterprise Center at the UCR campus. A few other reminders. There's a short survey that we would like for you to take at the end of the lecture series. If you haven't already gotten the survey, please pick it up on your way out and fill it out. That will really help us for planning for the future. Also, if you've not yet received a t-shirt, we're giving out free t-shirts and we still have some left. They seem to be very popular, so please, if you already have one, don't take another one. Finally, I'd like to introduce our speaker, Carl Krainer, tonight. But just a little bit more bookkeeping. As you see from the 5 chairs up here, after Professor Krainer gives his talk, we're going to invite the other 3 panelists up here, and we will have a discussion. So any questions that you have, hopefully the panelists will be able to answer. So Dr. Krainer is a distinguished professor of philosophy and a faculty member of the Environmental Toxicology Graduate Program here at UCR. He graduated cum laude from Mathematics and General Studies at the University of Colorado and received a doctorate from UCLA. He also received a master's in the studies of law at Yale Law School. He's written widely on philosophical issues at the interface of law, science, and philosophy, and leading to the publication of at least 6 books as well as many articles. And he'll be mentioning one of the books in his talk tonight. In addition to what's in the program, you'll see that I'd like to mention that Dr. Krainer has served on several scientific advisory panels, such as the panel for Proposition 65, the Electric and Magnetic Fields Program, the Nanotechnology Program, and the Scientific Guidance Panel for the California Environment Containment Biomonitoring Program, and he's still currently serving on that one. He's also served on the National Academy of Science panel to the Czech Republic, and on the Institute of Medicine panel, Valuing Health, Cost Effectiveness Analysis and Regulation. So Dr. Krainer's talk tonight will be implications of genetic modifications in humans and other organisms. So please join me in welcoming Professor Carl Krainer.
Oh, I'm on. Thank you very much, Francie, for the introduction. The title's slightly misleading. Well, let me say, first of all, great to see all you here. A number of my friends from the sciences are here, so that'll keep me honest. I'll try not to go beyond my pay grade, which would be easy to do. The title is slightly misleading because I'm mostly going to talk about humans, and what I'll talk about, I don't know to what extent CRISPR can address these issues, but we can sort some of that out in the question period. So let's see, oops, okay. I have to figure out which direction to go here. So CRISPR is an acronym for clustered, regularly interspaced, short palindromic repeats. I'm not going to explain that. We have 3 scientists here that can do a much better job at telling you. I'm very interested in the palindromic part, but I won't go there. But what it is it provides a faster, easier approach to modifying DNA that will change how basic science is done and how we think about treating diseases. It's already used in a variety of circumstances. It's used in industrial processes where bacteria cultures are used. It's used in science lab for precise changes in the genes of various biota like fruit flies, fish, mice, plants, and even human cells. And the interest is that scientists can modify specific genes while sparing all others, and clarifying the association between a given gene and its consequence to the organism. How might we use CRISPR for medical purposes? Well, one way is to think about treating genetic diseases. Scientists, by replacing the mutant form of a gene with its correct sequence in adult mice, cured a rare liver disorder, with a single treatment. I think about cancer a lot because I've done a lot of work on carcinogens. They can make specific antibiotics that target only gene-causing bacterial strains while sparing beneficial strains. They can use it to make white blood cells resistant to HIV infection. In treating HIV with gene therapy, here's a real kind of advantages. There's either a lesser or no risk of rejection of newly created cells because they come from the patient. So the patient wouldn't be rejecting foreign cells out of the body. And they will prevent preventing the production of certain genes as part of the therapy adversely affect a patient's health elsewhere. I think that's one of the questions that needs to be addressed as these things develop. Now, I've been asked from time to time to contribute to volumes, or even a volume I did. There's a certain temptation to think for every gene there's a cure. For every bad state of affairs in humans, there's a gene that will cure it. If you want to jump higher, get a jumping gene. If you want to be smarter, get smarter genes. If you want to do math better, get math genes, and so forth. It's not really clear how many monogenetic diseases there are. Here are 7 or 8 that are listed by the World Health Organization. I think Martine said that there's a much larger number that might well be single gene diseases. And it does seem to me that one probably should be careful about this because we are a biological entity with all kinds of things that impugn on various organs and genes in our bodies, and our genes control various things. So how much there will be one gene, one effect, you may want to ask us about it and talk about it. One of the things that can happen with CRISPR, as I understand it, is that perhaps cutting the target gene may be wobbly. Researchers at Harvard discovered this just in March, and it was reported in the journal Science. A slightly older version of CRISPR didn't cut precisely. They said it was wobbly. I don't totally know what that is, but we have people that can tell us. And there was a concern they could be off-target and create off-target mutations or interfere with proteins, producing adverse effects. So those are some of the things one wants to look at as this technology is developed. Of course, tinkering with the genes, I guess that's not a technical term, modifying genes, I should say, invites the idea of eugenics. And I want to distinguish a couple of different kinds of eugenics. One is having the social intention or maybe a legal requirement to protect or improve the gene pool. I'm largely going to ignore that, and I'm certainly going to ignore the odious, I love these words, the odious, vile, and discredited social policies of Nazi Germany. They should be properly discredited. However, the United States is not free from critique. A U.S. Supreme Court decision in 1927 upheld gene modification for eugenics. Virginia, and this was a case of Carrie Buck versus John H. Bell. Virginia and many other states had compulsory sterilization laws for the intellectually disabled to foster eugenics. Carrie Buck's mother, with a mental age of 8, with a record of prostitution, had 3 children. Now I think, that far back, the history of that might be very interesting. Did she really have a mental age of 8? Did her parents not let her go to school? What was going on? But anyway, she was judged to have the mental age of 8. She gave birth to 3 children, Carrie Buck being one of them. Carrie was committed to a state colony as feeble-minded. Again, I think you want to think very carefully about what feeble-minded amounts to. She reached the 6th grade and eventually had a feeble-minded child of her own. Because of this, the state colony ordered Carrie's sterilization, but her guardian appealed that order. And that order made the U.S. Supreme Court. And I don't want to talk about highlights of that decision. These are really low points of that decision. The court said at least the following 3 things. Compulsory sterilization of the unfit, whatever that is, including the intellectually disabled, for the protection and health of the state, did not violate the Due Process Clause of the U.S. Constitution. So if Virginia passed this law and it was implemented, this passed due process. A person, by being sterilized, did not have their due process rights violated. We have a constitutional provisions that you can't take the rights of a person or their property without due process of law. Being sterilized did not violate that, according to the U.S. Supreme Court decision written by Oliver Wendell Holmes. He went on to say, Carrie Buck's welfare and that of society will be promoted by her sterilization. And then near the close, he said this: 3 generations of imbeciles are enough. So the U.S. is not free from eugenics ideas either by law or policy. 15 years later, in 1942, another case made the U.S. Supreme Court. In 1935, Oklahoma had passed a law authorizing the sterilization of habitual criminals in the criminal justice system. And just before 1942, they sought to force the sterilization of Mr. Skinner, who was a habitual criminal. He was one of these people they wanted to weed out as an unfit individual. This case also made the Supreme Court, and to its credit, it did find that this law violated the Equal Protection Clause of the U.S. Constitution. Why was that? Well, this law was used against kind of ordinary criminals that steal and burglarize and take valuable things in that manner. But if you're a white-collar criminal, you went off scot-free, and the court said, got to treat like cases alike. You're a thief, you're a thief, and those white-collar criminals ought to be sterilized. Well, you would imagine that there was probably a lot of pressure not to sterilize white-collar criminals, captains of industry or the local railroad or whatever. But those folks that, burglarized houses, that's okay. Supreme Court said you couldn't do that. Those were a couple of examples, and especially in the United States, of eugenics by social policy. It's somehow built in either to the legal system or to kind of social consensus. But what I want to talk about the rest of the time is having the consequences of modifying the gene pool and how that can come about. The idea of agency is broader, including individual choices. So if you do something and I do something, another person does something that modifies the gene, this could skew the gene pool if too many of us do it, and that's the kind of thing I'll talk about. So the agency is broader, including individual choices, and it's not necessarily deliberate state or social programs. The motive is irrelevant. It's not necessarily an intention to improve the gene pool. Yet, collective good or bad consequences affecting the gene pool may in fact result. When Francie asked me to do this, I said, well, you know, you're going a bit beyond my pay grade, but let me see what I can do. And then I did edit this book way back 24 years ago, hard to believe. And I draw heavily on, for this lecture, on work by Diane Paul and Greg Kafka, and bits and pieces here and there of some things I've done. So CRISPR is a tool. How should it be implemented or used? It's a scientific tool. Well, I thought just for contrast, I would remind you that hammers are tools too. Hammers are just much less complicated than CRISPR. And I put up some pictures here of complicated tools that we've dealt with over a period of time. You can, there's a story there in every one of those about how it started, what improvements had to be made, downside effects that they discovered, benefits from it, how they balanced those in the end. I'm not going to go through all that, so you can relax. But I do want to talk about this one because it is tied in with the lecture in an important way. In the 50s and 60s, some of us, I confess, are old enough to remember our parents taking us to get new shoes. And at the shoe store, they had this wonderful little machine that you could look into, and you look down at your feet and you saw the bones. Ooh, that was neat, wasn't it? They were selling this idea. Well, the idea they also sold at the time was that you could treat tonsillitis with radiation. And some people had their tonsillitis treated with radiation. The main person I'm going to rely on, who wrote an essay for my anthology, was Greg Kafka. Greg Kafka received radiation for his tonsillitis. It caused lymph node cancer that led to bone cancer, and he died at age 47 because of this tool that wasn't fully understood what it could do. So in a way, I think I'm dedicating this to Greg. He died some time ago, and it was a good friend. Here's somebody that recently realized that, well, maybe we need to be a little more careful how we use these tools. This is Mark Zuckerberg in testimony before Congress. We didn't do enough to prevent this tool, Facebook, from causing harm to the American public. So what collective steps should we think about and take to forestall various potential undesirable consequences from the use of CRISPR? Well, Martine's already presented an international summit on these issues with the involvement of these agencies from different countries and a couple of our own. So I think about CRISPR, there are a couple of different tasks. There's a scientific task, and then there's a social task. And the scientific task is we should carefully vet by the scientific community for potential adverse effects. The National Academy of Sciences, that's begun obviously, the Institute of Medicine committees, and at some point I think mostly about using CRISPR on humans, but could also be used on agriculture, as Carolyn presented. There it should undergo careful review by the Food and Drug Administration about when it could be used and when there's the very limits and so forth and so on, and EPA particularly for agricultural kinds of purposes. But since it'll be easy to use, what safeguards are needed on who can use it, what can it be used for, and how should it be used? So I want to talk about the social effects, social assessment of some individual effects and what they could lead to. And they fall into 4 categories: collective imbalances, maldistribution of competitive goods, social inequality, and modifying collective consciousness. What I mean by this is that the cumulative effects of individual decisions about either traits of offspring or traits, changes we might make, even in adults, I suppose, can distort a desirable distribution of characteristics in the general population. So consider sex selection. I don't know if CRISPR can be used to actually select the sex of a child in utero, but if it can, there are potential downsides. And I was fortunate, the Washington Post was aware we were going to have this discussion a couple of weeks ago, and they ran a story on the maldistribution of sex selection in China and India. There are 70 million, got that? More men than women in those 2 countries. This distorts labor markets. It drives up saving rates in China, drives down consumption, actually artificially inflates some property values, and increases violent and probably sexual crime, trafficking, or prostitution in some location. And here's the graph, as it were, that was in the Washington Post, with the gap, the width of the graph part there, the black and the brown, indicating how many more boys or males than females there are. They do anticipate that it'll go down over time, but they don't show it disappearing. So those countries are going to have a maldistribution of gender for some long time. So should society discourage, even prohibit sex selection by parents, except maybe for sex-linked diseases? Should potential collective problems override individual autonomy and choices in families? We could imagine collective imbalances in things like choice of psychological or social traits. Suppose you want your children to be better off in certain ways. You might want to create children with hyper-competitive tendencies so they will rise to the top in our society. Individually, they might function quite well and tend to be successful. But what are possible collective consequences? We may have too many generals. Will there be constant conflict among the A-type personalities that have been ramped up by the genetic choices of their parents? Will there be discouragement of others from putting forth greater productive efforts because it's wasted? You've got all these smart people out there that have been ramped up genetically, and how am I supposed to compete with them? Okay, one might alter genes for longevity, delay the effects of aging, and prolong the human lifespan. Well, what about this? Is this even a good thing at the individual level? And again, the Washington Post anticipated this lecture because they ran a story just in the last couple of days on a scientist from Australia named David Goodall, who said, "I greatly regret reaching 104. I would much prefer to be 20 or 30 years younger." And interestingly, I don't know if you read the Post or the New York Times, he ended his life today at 12:30 a.m. Switzerland time. He's done. And people kept asking him, "Do you regret it? Do you have the slightest sliver of regret?" His answer was, no. So it may or may not be a good thing individually. You might think, well, yeah, it'd be great. Maybe I can hold the world record for the longest living human being or something. But do you have good health along with that? Or have all your friends died or whatever? But will longevity increases be collectively desirable? So we've got individual. Is it a good thing for the individual? Is it a good thing for the collective? Will it exacerbate current trends? Will it impose a costly economic burden because of the increased population? Already we know in the United States that as we're growing older, we don't have enough working age people to well support some of the social programs that people are getting older are going to need. So this would only exacerbate those kinds of problems. Should we extend longevity, but maybe limit the right to reproduce? Say, okay, Craner, you can You can live as long as you want, but you can't have any kids or any more kids. That's it. We don't want you adding to the population problems. Or could we retain the right to reproduce, but not permit modification of genes for longevity? We may begin to face those things if an easy-to-use genetic technique like CRISPR can produce these modifications. So what's going on here with collective imbalances? Individual choices using CRISPR selection may change the frequency and distribution of traits in the population. So autonomous or individual acts are not harmless. How should this developing technology be used? So should there be social constraints or not? If not, will some of these issues be manifested? If so, will some object on parental freedom grounds or autonomy grounds? You're keeping me from making my children smarter or more competitive or whatever it might be. Then we have competitive goods. Thomas Hobbes, many of you may know the very famous slogan from Hobbes. He was a political philosopher of the 1600s in England. And his famous slogan is he urged that, or he asserted that life was solitary, poor, nasty, brutish, and short. Okay? He might have been interested in longevity genes, I don't know. But he also thought that the human life mimics a kind of race. And the main goal is to be foremost. So to be outdone is just misery. But to outdo others, that's terrific. And so he saw this as a very competitive world and was thinking about that. But competitive goods are positional goods, things like status, honor, victory, ranking above others on some kind of comparative dimension. But, of course, not everybody can rank first or high up. Somebody's going to be disappointed. Positional goods, of course, can be intrinsically valuable for their own sake. You may want to try your best to be as good as you can at something. But they're also important means to other ends. They're extrinsically valuable. They can lead to more money, better access to other persons, or higher rank. So we could imagine gains in competitive goods, like suppose we could take CRISPR and make our children smarter, friendlier, or lovelier. This might make their lives and our lives go better. Might not, too. But what are some collective effects? Well, greater intelligence is an important individual good. We probably have a room full of greater intelligence here. And it's an important individual good, both to the person who has developed their intelligence and extrinsically because it leads to other good things. But if too many parents using CRISPR for their children to increase the intelligence opted for greater intelligence, this will not likely confer competitive advantages. Why is that? Well, if everybody's getting smarter, you're not going to outrace anybody. You're going to be even with others who had the same kind of genetic treatment, if indeed that was widespread. Okay? But you might ask, well, is a smarter population better off? Imagine a Dr. Spock, a country full of Dr. Smocks. Is that a better country than the one we live in? Or do we need people to do some of the other tasks? There's also the fairness issue. If some do not choose the best intelligence genetically, and it might be for a variety of reasons, moral, religious, cultural, or personal reasons, is this unfair to their children? Suppose you don't choose that and later, children sometimes surprise you, the things they remember from their childhood. You did this to me on January 12th, 1983, and I've resented it ever since. Well, what if you didn't choose the best intelligence? Will it pressure parents to compete on this particular dimension? But suppose you could use CRISPR, but maybe it's not totally free from accompanying physical or mental damage with the attempt to improve intelligence. Is that a risk people should take? Is it a risk we should let people take? Should we, how much should there be controls here, and so forth? So what occurs here from some of these examples is what might be individually rational and desirable from a person's point of view may not be socially desirable. This is one of the major contributions Greg Kafka made in his research over the years. How do we address that? Well, we could change the incentives, perhaps by enforcing a ban on certain choices. We might foreclose certain genetic enhancements of intelligence or other competitive goods. Would that be a good thing? And isn't correcting deficient competitive traits different from enhancing normal or natural capacities? Well, this doesn't seem competitive because these competitive traits tend to be continuous. They're not like on-off switches. You either have it or you don't. But rather there's a graded scale from various degrees perhaps of deficiency, if we want to use that term, to substantial enhancement on a scale. But there are small steps from one to the other. It's not like you're totally without or totally have the enhancement. Also, the extent of advantage or disadvantage depends on the extent to which others possess them. So what's the underlying problem here? Well, Greg Kafka pointed this out. If normality and deficiency in competitive goods are characterized according to social averages, the main arguments against genetic enhancement of the normal also apply to genetic enhancement of the deficient. You're just going to make some minor changes in the average and probably move the average either up, but maybe not change relational positions within it. But we already permit parental enhancement of various kinds: preschool education, cultural enrichment, travel to foreign countries, even collective enhancement, things like Head Start. So we think that's a good thing. So Greg actually suggests a couple of different possibilities here. One would be a really radical path, which I think he doesn't believe we would go down that path. But maybe we've been too permissive in the past about parental enhancement, and thus should limit it along with genetic enhancement. Does that make sense? Or on the other hand, we might tolerate genetic enhancement, correction of various deficiencies that we can identify, if there are, as he puts it, provisions to deal sufficiently with concerns about fairness and equality. I think that's terribly important. And next. So, if a child suffers from a serious disease that CRISPR could easily, but we'll have to ask Martine about this, expensively correct, what's it going to cost to correct some disease, should all have a fair shot at correction. Think of this as something like equal protection from genetic harm. This is a kind of, whatever it is, a picture of the distribution of income in the United States that I use with respect to a class. And you'll notice that from here over, 71% of us have earned roughly less than $50,000 per year. It's only a fairly small percentage that earns more than that. And of course, we have the top 1% that seem to have no limits. So we have a substantial distributional problem already in the country. How would easy genetic fixes feed into that? Who should benefit from CRISPR repairs for serious deficiencies if it's permitted? Well, this is a graph from the Center for Budget and Policy Priorities, and you'll notice that 10%, the top 10% of the public controls 50% of the wealth in the country. However, the top 10% control 78% of the wealth. So this gives you some picture of the distribution of income and wealth in the country. And this was as of 2017, so it's fairly recent. So should we have unfettered capitalistic eugenics? Determine CRISPR by use of, whoops, what happened here? Oh yeah, determine CRISPR use by wealth. So here we have only the well-off would have access because only the well-off might be able to afford this, but the scientists can give us some idea of the entire institutional setup that it would take to use CRISPR to take advantage of some of these things. The scientific technique itself is simple and apparently, Francie tells me, very inexpensive, but by the time you build hospitals and doctors and so forth around it, may well drive up the cost. So who can benefit from this? But there's another alternative, not CRISPR use by wealth, but one in which everyone with a serious disease that CRISPR could easily, maybe expensively, correct, would have a fair shot at correction. And here you might think of genetic treatment as somehow equally open to all, resembling perhaps an improved version of organ transplant. That would raise perhaps what can we afford if it is open to all. But I use this line to suggest that it doesn't matter what income group you come from, the lowest, the next lowest, the mid-range, the upper middle class, the very upper middle class, or the quite well off. You should have the same chance of correcting your genetic diseases if they're really serious. That's a much different picture than saying only the top 1 or 7% can get access to CRISPR. Then the last point that I want to make, several slides, is that introducing an easily used genetic tool may change how we think about ourselves, our sense of consciousness. So those with religious views may think of themselves as having a soul infused by God and their lives influenced by it. And here we have the wonderful painting from Michelangelo's Sistine Chapel. Absolutely wonderful, igniting Adam to life. Okay? Genetic advances may also influence, but they're going to influence how we think about ourselves and the world, very likely. In a genetic world, if genes determine our identity and we have the ability to manipulate them, we may have the power to play God, to do things that we maybe previously attributed to a God. Will this be seen as monstrous fooling with the foundations of our own existence? In such a world where these genetic tools are readily available, would we view parents as more responsible for their children as our achievements? I mean, already I think, very often parents are, I don't know, rightly or wrongly given credit for how their children turn out. Oh, you must be very proud of what your child accomplished. Well, yes. Whether it's deserved pride or not is another question. Okay, but if we can modify the genes, will this be exacerbated? Okay. On the other hand, would we become less tolerant of diversity if genetic techniques were used to foster normality? We had some conception of normality that there were social pressures to promote. Would genetic successes increase the faith in technology to provide solutions to many problems? You might think of these as science fixes more than more humane problems. Suppose your child came home from school and said, you just can't do this algebra. Would you send me to the genetic doctor to give me a better math gene or give me a better English gene so I can understand this blasted poetry better? And if your parents refuse, then what's going to happen? Dad, Mom, I need a better math gene. Okay? With the genetic power to shape the species' biological future, will we feel an awesome, overwhelming sense of responsibility, or maybe be at sea about what should be done? Years ago, the existentialists, the philosophic existentialists, mostly from Europe, probably terrified people when they suggested there is no God, and thus there are no external norms or purposes as part of the universe. I always thought that was kind of a bad argument, even as an undergraduate and certainly in graduate school, but I'm sure there are people that would worry about that. But if we achieve vast power to modify our genetic heritage, will we see ourselves, humans, not God, not chance, not nature, as responsible for humans' future? Will vast scientific power, genetic power, lack of clear standards, plus inescapable choices be a prescription for, as Greg puts it, a tormented collective soul? I'm not sure quite what a tormented collective soul is, but that was one of his worries. Or you might say operationally, for intense political conflict, much more likely. Or will we be unusually careful and thoughtful and cooperative in developing responsive social policies to these new technologies? That's my hope. Thank you, and now we'll have a panel discussion. All yours.
Yes, yeah. Thank you, Carl. That was actually a great talk. I'd like to briefly introduce each of the three panelists from the previous talk. Martin Garcia Castro, do you want to come up? Martin is a professor in the School of Medicine, and he works on early development issues, particularly in the neural crest. So Martin gave our first talk, which was The Hope and Threat of Human Gene Editing. Our next panelist is Professor Carolyn Rasmussen from the Department of Botany and Plant Sciences, and her talk was on feeding the world from Mendel to CRISPR, and she's a plant biologist. And our third panelist was Ansel Shao from the Department of Microbiology and Plant Pathology, and the title of his talk was Lego, Language, and the Life of Bacteria, and he works on bacteria and bacteriophages, particularly in the gut, and how those could be modified. So what we'd like to do is have you come up on either side. There's microphones on the sides, and ask questions to anyone in the panel. I will repeat the question for the audio recording, and then we'll let the panelists answer the questions. All right? All right. So we'll start off over here.
Hi. So I didn't really get this from you. If you were saying the example of, I want my kid to be smarter, have more intelligence by giving him CRISPR, how would, can you physically put CRISPR inside someone and choose what you want him to get? Like what you want to give him, make him smarter, change anything in his body? But if so, is it successful every time?
Okay, so that was a very good question, sort of a technical question. Is it really physically possible with CRISPR to make somebody smarter? And I think that was the basis of it.
Yeah, and basically change anything in his body.
Yeah, can you change anything in your body with CRISPR? I'm going to pass that off to Martin.
Thank you very much. So you can change many things in your body through CRISPR, many different genes for functions. But at this moment, to make you more intelligent is far-fetched. I think we are very, very far from that. We don't understand enough about the function of the brain to achieve that. But I think the more important question is whether we should do it, because we will have the capacity at some point. And as you could see from Carl's excellent talk just a second ago, 20 years ago, he put a paper or a book that essentially deal with the issues that we are confronted with today. And I think in that way, you should be the one that answers that and not just puts it out there. You have a microphone.
Yeah, I guess I would say, in answer to that question, I wrote a little essay on, as I told Francie, this is, in a way, it's above my pay grade, but I did write a little essay on, the human body is a pretty complex thing. And very often what things we can do, characteristics we have, are a function of multiple genetic and other contributions. And so it may be a long time before we're capable of trying to change all of those pieces so that they work together and they come out right. I would worry that they may not come out right.
Okay, all right, thank you for the question. We'll go over to this side.
My name is Vishant, and I'm a first-year undergraduate majoring in bioengineering. Already out there's a group that calls themselves the biohackers, and they're already out there modifying themselves. Just last October, a now famous biohacker named Josiah Zayner injected himself in the forearm with a specially modified CRISPR to grow larger muscles. In fact, he is the CEO of a company that makes kits that allows anyone to get their hands on CRISPR. I was just wondering your thoughts on this issue and the consequences for Zayner and others like him that have already started editing their genes because of widely available CRISPR. Is CRISPR going to become like a controlled substance?
Okay, so great question from a bioengineering student. I can see you're sort of planning your doctoral work already. The question was about biohackers, a guy named Zane, and he's CEO of a company who is claiming that you can do CRISPR on anything, and he's claiming that he has already CRISPR'd or hacked into his genome and made bigger muscles. So the question is this really real? And is this going to be available to people? Is it possible?
Well, he's injected himself. However, he has not seen results yet. But others like him have started injecting themselves with CRISPR, seeing if they can actually produce these results. My question is, will CRISPR become a controlled substance?
Okay, yeah, sorry. So the question is, so he's already been injecting himself with CRISPR plasmids, as have others. And the question is, if this stuff works, or even maybe if it doesn't, will this become like a controlled substance? So I will pass it off to you guys.
I should think if it becomes too easy to use, yes, you probably ought to. Well, you may not be able to control it. That's point one. Point two, there probably, this raises other kinds of problems, economic market problems and so forth. But you may want to have it used only in conjunction with an appropriate physician so that this is less likely to happen. But it's maybe so easy to use that all bets are off.
I would like to comment a couple of things in this regard. One of them is that it's a major concern that people would use it without truly understanding what they're doing. And that goes beyond CRISPR. One of the major players in the biohacker movement just died yesterday. He had developed a herpes vaccine. He injected himself, just like Asaya did with CRISPR, and he died yesterday. And so I think this is a concern that a lot of scientists share. But this technology is very powerful, very easy to use, but it requires a deep understanding. So it is very preoccupying to think that it will be used in a garage, in any house. How can you regulate it when it is so cheap? Mm-hmm. It's very difficult. At the same time, I have to say that fortunately, until now, it seems that the kits that are being sold by Isaiah are not working. So the same institute founded by Jennifer Doudna, one of the discoverers or founders of the CRISPR-Cas9, tested it. They bought his kit, and they relayed information that fortunately is not working. So he's selling $150 kits that are not that good.
Okay.
So I guess we can say this is a new type of snake oil, right? Some things don't change. Okay, on the other side then, please.
Hi, so my question is, do you guys think in the future people may abuse CRISPR to become more powerful than everyone else and like cause havoc?
So the question is, do you think people in the future will use CRISPR to become more powerful than everybody else and to cause havoc? Carl? That seems like, since I don't know what the power gene might be, I'm going to pass it off to you.
Well, I think there's a lot built into the word power. It doesn't mean they're going to be Arnold Schwarzenegger and be able to pump a lot of iron or something. But they might try modifying intelligence or whatever. That's probably a very complicated set of steps to do. And maybe, will they be tempted? I'm not a psychologist or a sociologist, but the answer would be probably yes. Will they succeed? I hope not.
Thank you.
All right. Over on this side.
Hi, my question is how soon before we start seeing products like drugs or food that have been grown using CRISPR in like common marketplaces?
Okay, so the question is, how soon will we start seeing products or food that have been, where CRISPR has been used on them? I think I'll pass that one off to Carolyn.
Okay, I think this is working, maybe?
Yeah, it'll pull off probably.
Okay. Oh, all right. I have the power. Can you hear me? No,
A little closer, maybe.
closer. So it turns out that the USDA has determined or has given a statement that CRISPR modified foods, crops, are not regulated are not regulated in a way that is different from the regulation of, and maybe you all will remember this from my talk, are not regulated differently from foods that have been or crops that have been altered by radiation mutagenesis, for example. So, but to answer your question, I don't know if there are commercially available crops that have been altered using CRISPR, but if there aren't right now, there will be. Oh, I'm sorry, I'm completely wrong about that. No, there are certainly commercially available products that have been, CRISPR has been used. Yeah.
So I think, Carolyn, you said that those crops do not have to be labeled as GMO? Is that?
They're not considered genetically modified organisms.
So that means we would be able to be buying food in the supermarket that could have been modified by CRISPR, not necessarily a bad or good thing, and we wouldn't necessarily know it. Is that true?
That's right. It's not, there's no labeling or anything like that.
But that probably will pass away when they start to regulate CRISPR modified, right? And they put a new label. I mean,
Possibly, yeah.
There is a point about GMOs. I only know a little bit about this, so that may be outside my pay grade as well. But I think that the USDA did not require the GMO labeling, right, on food crops that are now used. And I do know that there was some litigation, I think, in Vermont or Maine, where dairy farmers that wanted to produce non-GMO milk were not allowed to do so. They lost that fight, I believe. And so it's very difficult to find out if anything is GMO now. They're neither labeled nor are they allowed to be, or non-GMOs allowed to be labeled in some jurisdictions at any rate. But that's kind of complicated, and I don't know where it stands right now.
So, yeah.
Well, I'd like to say that I'm not familiar with that particular case, but I would like to just state for the record that the National Academy of Sciences, as well as a group of other people, have stated that genetically modified organisms are not more dangerous than non-genetically modified organisms. And there's a long history and a lot of data to support that.
Yeah.
Just to underscore this, so for example, 93% of the soybean oil used in this country is genetically modified. And soybean oil is about over 60% of the vegetable oil we use in this country is soybean oil. That means most of the vegetable oil in this country is genetically modified and it's not labeled as such. So that's happened already even without CRISPR. So another question?
Yeah. I'm just wondering if, because it seems to me from what I've been in the last lectures that I've heard, that it really does take a whole panel of scientists, or I mean lots of fields of scientists, to integrate all of the things that CRISPR can do. So I'm wondering if there's some sort of reality timeline, not people injecting themselves to try out things, because people, I mean, there are hackers, and it still doesn't seem to bother a whole lot of people. You know, we still all use our computers and stuff. There are going to be people trying these things, but in reality, because we still have a lot of research to be doing on this, what is a timeline when you see any of this stuff being actually brought to the fore where it affects society in a really obvious way, or a palpable way? Because obviously a lot of people are not going to benefit from, still a lot of people don't benefit from things that we can do now. So all of you are working on, integrating all the possibilities of this, all 4 of those possibilities. But what do you see as a timeline when it will actually be integrated and useful in a way that will benefit people in general?
Okay,
That's kind of a general question, but, this is a pretty specialized field right now with scientists and not available to all of us.
So the question is, what's the timeline on
Timeline,
this? So
basically,
it's obviously very complex. There's 5 of us up here, and we're still talking about a lot of stuff. Any idea in terms, if anyone wants to comment? Ansel, why don't, since you haven't had a chance to answer anything yet, we'll throw this one to you. What do you think the timeline is on some of the first applications in terms of CRISPR, let's say in terms of human health? It's already been used in food. We already heard about that. But let's limit it to human health.
Okay, so I want to preface this by saying that I do not use CRISPR in human systems, but I think that what you're going to see in terms of at least clinical or therapeutic approaches in humans, if it clears certain regulatory hurdles, I think that will begin in relatively short order. But I do want to point out that, when we're talking about biohacking, people trying to use CRISPR in their garages, it's really a black box in many ways, right? In terms of what genes cause what traits or what phenotypes, especially in an organism as complex as humans. And so this idea that somebody in their garage is going to figure out, well, it's this particular gene that's going to give us this trait, and then they're going to order this CRISPR kit and do it, I think is a bit far-fetched. And it's probably, there are many reasons why this kit is not likely to work.
I'm going to just throw out something out there, since Ansel works in the microbiome in the gut, so those are the bacteria and bacteriophages in the gut. You can, CRISPR, as we heard, came from bacteria. One could easily modify the bacteria in your gut with CRISPR. So in this case, we're not modifying the human genome, but it would be sort of an alternative to taking a probiotic. This would be a specifically engineered bacteria to either help ramp up your metabolism or produce a certain vitamin, et cetera. Could you give us a timeline on that?
That, I think, is, in terms of a verified product entering market, that might actually take even longer than some of the potential therapeutic approaches in humans. Because while we have been able to identify, okay, this particular gene or this particular sequence may be responsible for some disorder in humans, in bacteria, that's not really well understood. So we have hundreds to thousands of different species of bacteria, and understanding how those bacteria interact and how all of those genes interact is a very, very complex question. So while it is easier to modify a bacterial population and take it as a probiotic, in terms of getting it validated to the point at which it really works, it might even take a little bit longer.
Okay, so CRISPR not coming in our Dannon yogurt anytime soon.
But if I may add, there's already some approaches that will take advantage of this immediately, right? So there's some therapies for leukemia, for instance, for very heavy leukemia that initially relied on other type of gene editing, and today they think they will deliver it with CRISPR modifications in a flash. And so those are a reality. And in some of these diseases in which we know of a specific gene that is responsible, delivering the modification might not be that far-fetched. Those will occur in a relatively short time.
I was hoping for a little bit thinner biome to help out with some of my, so I had to run less.
Yeah.
Thank you.
Over on this side, please.
All right, thank you. As it was touched on earlier in the lecture, there may be some economic ramifications brought upon our society if, per se, the longevity of human life is able to be increased. That being said, do you think that it is viable to introduce these types of positive gene alterations into our society, given that society has, throughout the years, evolved to adapt to our increasingly technological age? Does it seem more sustainable to slowly introduce this technology to the population and have it trickle down from the wealthy elite to lower socioeconomic levels, or introduce it at a later time when it is relatively inexpensive and available to the masses?
Okay, that's a great question. I think there might be more than one question in there. First started off with longevity. And I think the question there was, is this a good idea to do it? And what are the social and economic implications of altering one's longevity? And then the next question was, would it be better to hold off on letting it be used in society until we have better means to make it easier and it's certainly cheaper and more accessible? So, Carl, we'll pass off that regulatory question to you.
Well, it seems to me that I would argue for holding off on that because we have already in this country enormous maldistributions of both income and wealth. And insofar as that gives people competitive advantages or allows them to pass on things to children or whatever, it seems to me that socially and economically and on grounds of justice, it would only exacerbate what we already have. So I would urge that we wait until we can provide it much more generally across age groups, whether it's correcting for Huntington's disease. A poor person with Huntington's disease is just as bad off as a rich person with Huntington's disease, and each should be corrected if CRISPR will allow that. But you can bet that if we don't pay some attention to this, it won't be.
I'm going to throw a wrinkle in this. One could maybe make the argument, since CRISPR, at least in the lab, is relatively cheap, and let's assume you could identify the appropriate gene to give you the appropriate advantage, and if it is actually cheaper, maybe we're all thinking about having the rich having accessibility to it and exasperating differences. Maybe this could democratize stuff, and having people with lower resources being able to then enhance themselves, and then you could close some of that gap.
Mm-hmm. Well, the preeminent theory of justice in the 20th and into the 21st century was written by John Rawls at Harvard. He has now died, but he had a couple of ideas that were terribly important. He thought, if you remember my picture of the silos, he thought that the idea of fair equality of opportunity should apply no matter what socioeconomic group you come from, no matter what geographic area you grow up in. You grow up in a poor town of rural California or Colorado or Mississippi, and you have a child with same talents, abilities, and motivation as somebody from Beverly Hills. They should still have the same shot at getting to Harvard Law School or wherever it might be. So there is the notion of fair equality of opportunity. A second principle that he introduced was what he called the difference principle. And he made the argument that unequal distributions of income and wealth were fine up to the point where those who are well off start using their advantages to damage the life prospects of those under them. So this theory has a couple of guidance principles that would be very helpful in thinking through some of these issues.
But what if one wanted to use CRISPR? Let's say you're on the lower end of the resources, and now you have CRISPR available, and that will bring you up a little bit closer to what the people with the resources have.
I think that should be applauded, yeah.
I think we're over on this side.
Hi, my question is, could CRISPR be compared to a drug like steroids?
Could CRISPR be compared to?
To a drug like steroids.
Oh, to a drug like steroids. Martine, do you want to answer that?
You want to answer that?
You want me to answer?
Yeah,
I can answer this.
because
Okay.
I think I will answer very.
So I think steroids, and I'm a plant biologist, but steroids have a very specific function in the human body, whereas CRISPR instead is a technique that we can use to target specific genes. And so the potential outcome of that is much broader than, for example, what would happen if you treat a person with steroids. So it's just the potential effect would be much more variable.
So I'll just throw something in there. So, athletes, bodybuilders, they take steroids. Obviously, men have more of these steroids than women. That's why they're able to build more muscle mass. The notion of, so steroids do a lot of different stuff. In a way, I mean, they're going to affect every cell in the body. In a way, one could even make the argument that CRISPR could be more specific actually than steroids. And then the notion of, well, is it fair if you naturally have lower steroids, why can't you just enhance your steroids a bit? Same with people when they're aging. When they're aging, our steroids go down. That's one of the, one of the main drivers of all the symptoms of aging. And if we could do something to, if we could take, is it okay for us to take steroids? There's a lot of health issues involved with that. That's the main reason not to do it. But let's say we could have some steroids that are actually, we could eliminate the negative side effects. Well, why not take some steroids and feel younger, right? And so then the question is, would be a similar question with CRISPR. Okay, good question. On this side?
Hello, I'm a fourth-year student here at UCR, and I'm hoping to research more into CRISPR and pharmaceutical uses for it for my graduate level research. And my question is, there are companies who are looking into using CRISPR-Cas9 and in conjunction with research on CAR T cells, which is supposed to aid in, I guess, just solving and diminishing cancer altogether, like how Novartis has created the gene therapy called Chimera. Do you guys see CRISPR being used in future biomedical uses, just in general, since it's going to be so accessible?
Okay, so the question from a fourth-year UCR student who's interested in going to graduate school and interested in working on CRISPR and pharmaceutical uses, he made mention of the fact that CRISPR is already being used in CAR T. CAR T is it's a chimeric technology involving T cells that is all the rage in big pharma, in the pharmaceutical industry, as a way of modifying and ramping up the immune system to treat cancer. So I don't know specifically people using CRISPR for it, but I'm sure they are, just because it's a very important technology. It's really being driven in a lot of different ways, being used in many different aspects, mainly with cancer. And in this case, particularly when you're dealing anything with cancer, the cancer patient, if they don't get the appropriate therapy, unfortunately is going to die, right? And in this case, you're also targeting the cancer. You're not talking about modifying someone's genome, right? So I don't know, I haven't read any specific paper about CRISPR and CAR T, but I'm sure they are doing it, because why not? It's just a really great way to add modifications into human cells. Martine, do you want to add anything else on that?
Yeah, I think there's many uses, current uses of CRISPR for health strategies and in pharma, from using them as an alternative to antibiotics to preventing disease. I mean, it's just rampant. It's being used wildly.
Over on this side?
Howdy, my name's Danielle Hamilton. I'm a fourth-year biochem student here at UCR. My question has to do with if you modify somebody's genes, say, to make them stronger or more competitive in some way, I'm guessing there's no way to detect that. Like, we can test for if somebody's taking steroids, right? But do you predict that perhaps somebody will add a modification to the edited gene region? I don't know if this is possible, like fluorescence, so that you can detect if somebody's gene's edited and maybe disqualify them from applying to a job?
Okay, all right, that's a great question. So another great question from a fourth-year biochemistry student. Is there some way that we can detect whether or not someone has used CRISPR? And again, the analogy is back with steroids. You can test their blood or actually their urine to see if they've been taking steroids. I think I'll try to answer this one. If we could be successful in an adult taking using CRISPR, so in contrast to an embryo, you only have one cell that you need to modify, or a couple cells in a blastocyst. In an adult human, if you're going to have an effect, you're going to have to modify many cells. You won't ever modify all the cells. And so just as with cancer, you often, when you're looking at what genes are driving a cancer, you take out the tumor and you take out normal adjacent tissue. And then you look and see what's in the normal adjacent tissue versus what's in the tumor, and you can see those differences, right? The same thing will be there for CRISPR, because you're never going to be able to modify every cell in an adult human. I don't care how good CRISPR is going to get, it's just not going to get that way. And you can definitely sequence the genome from a single cell. So you would be able to tell that some cells in the body had, whatever that modification is, had the change by CRISPR and other cells did not. It's not going to be as cheap as testing your urine for steroids, but you can still do it. Great question.
Thank you.
On this side?
Hi, I'm a junior from John Dunmore High School, and I was wondering if it's possible to edit our genes to produce embryonic stem cells instead of adult stem cells using CRISPR.
So the question from North High School is, can we use CRISPR to edit our genes? Can you repeat the question?
To produce embryonic stem cells instead of adult stem cells.
Oh, so can we use CRISPR to take like an adult cell and turn it back into an embryonic stem cell as opposed to trying to isolate adult stem cells?
No, can we like using our bodies, can we use CRISPR to like edit our body to produce embryonic stem cells instead of adult stem cells?
Can we use CRISPR to have our bodies produce embryonic stem cells?
Yeah, instead of
okay. Martine, I'm gonna, I have an answer, but I don't want to dominate here.
Go ahead.
Well, so we have very effective ways to make induced pluripotent stem cells that are going to resemble the embryonic stem cells. So it seems a little bit convoluted to introduce a gene editing when what you want is to express between 3 and 4 genes only temporarily to achieve that goal. So my prediction is that it's not a good use for CRISPR technology.
So just with the basic question of producing an embryonic stem cell, the easiest way to do that is you have a male mate with a female and you'll end up with a fertilized egg, and that's an embryonic stem cell. So we don't need CRISPR to do that.
I know, but like, is there a different way to do that, or is it just that way?
Well, so actually maybe another way to phrase the question would be, if we have this thing, as Martine mentions, of induced pluripotent stem cells, right? And so in order to do that, you add a mixture usually of 4 genes to take that adult cell. Doesn't have to be an adult stem cell, but take an adult cell and revert it back into an embryonic state. And currently we're using traditional recombinant DNA technology with that. But yes, you could definitely use CRISPR with that.
Actually, I mean, most of the time what people do now is to do just a short pulse of the presence of these proteins. That's a much preferred way, because then you don't modify genetically these cells. You don't know where the gene is going to land, etc. So you just want them present for a little bit to reset the cell into the embryonic state. So yeah, I think CRISPR is not well suited for that goal.
So nice question, nice question. On this side over here?
Hi, I'm a high school student taking AP Biology, and I kind of wanted to ask what someone already mentioned before about control, just to kind of clarify. Is there any kind of control, like currently on CRISPR, like who has access to it, and is there any laws or rules being passed about how to use it and things like that?
Okay, so a question from a high school student taking AP Biology. Are there any current regulations in terms of using CRISPR?
Yes.
I don't know the answer to that, but I think in the early stages of technology, it's important to think this through and nationally get Congress to act. This is something that doesn't happen frequently. But let me give you a story. Probably 8 years ago, I was at a conference at the chemical, whatever it is, the American Chemical Institute in Philadelphia. And the question of nanoparticles came up. And there was a speaker there from industry, and I don't know whether he was sincere or just being disingenuous, but he said, look, we're creating nanoparticles. We need some guidance. We would like the appropriate federal agencies to step in and give us that guidance, but they hadn't done it, and they never did get around to doing it, and so now we have nanoparticles everywhere without the guidance. So I think getting in at the beginning and attuning people to some of the pluses and minuses and things that should be paid attention to is terribly important, because in a vacuum, people will take advantage of it.
So I guess an analogy is, or another analogy is with cloning humans. The idea of with other organisms, you can take an embryonic stem cell and make the whole organism. You can even do this with induced pluripotent stem cells. It's completely banned in the US to do it with a human, right? I'm not sure about other countries though.
So there should be more push for control then, like soon. Okay.
And so there's no regulation. The answer is that there's no regulation at the moment, and that there's a lot of experiments being done that should be controlled. And there's a lot of efforts by the scientific communities and by some governments to try to regulate it. And it requires the involvement of the community, where we all discuss this and allow the general public to understand it, and then we can take decisions.
Okay, all right, over on this side.
I wanted to ask, do you think this technology's ultimate potential will be limited due to societal controversy?
The question is, do you think this technology will be ultimately limited to societal controversy? So like things that we're talking about now.
I didn't understand the question.
Well, so we've been talking about the technological aspects and how easy CRISPR is to use to alter a genome. But the question is that will it be limited because of these societal issues that we're talking about now?
You need a prognosticator for that. But it's probably good, again, it's probably a good idea, as Martine indicated, to think it through and see go back to the slide I had with the different kinds of technologies, and imagine the steps that have been followed with respect to the technologies. I had a picture of an old car, but we have a lot of regulations that weren't around when those first cars started. So we had to develop them for all kinds of things, the fuel that's used in them, the way the highways are built, and so forth. The earlier you can think those problems through, I think, the better.
For sure.
Okay. All right, thank you. Over on this side.
Hi, my name is Dana Abuabis. I'm a second-year biology student here at UCR, and my question is, can gene editing using CRISPR cause a new domain to appear on a polygenetic tree of life?
So the question from a second-year biology student at UCR is, can CRISPR be used to introduce a new domain on the tree of life? Okay, I'm going to let Ansel answer that one.
Yeah.
He's sort of an evolutionary guy.
Well, okay, so I guess my answer is going to depend on how you want to measure domains of life, right? So it's certainly conceivable that you could take a cell and heavily modify it so that it's now, has a very different catalog of genes available compared to what it was before. When we talk about domains of life, we often use genes that are single genes that are common to basically all organisms, right? And so it is theoretically possible that you could take, for example, a ribosomal RNA gene and edit the sequence of that to be something that looks quite different but still functional. And so at that point, you might consider that a domain of life, but really what that cell does or what that organism will do is going to depend much more on all of the other genes that are present, right? So in a very technical sense, I guess we could introduce new phylogenetic groupings of life, but in terms of the function, right, and in terms of the structure of that cell and that organism, that is technically much, much more challenging. You have to change a lot more genes in order to do that.
So we might also then have to change what we define as a domain of life, because now if it's done mainly genetically through a few key genes, we have to rethink that then, I think is what you're saying.
I just remember that there's big group of biologists called synthetic biologists, and they've been modifying genomes very heavily, and they've reached a point where they can modify a ribosomal RNA, where it codes for, the tRNA now codes for an amino acid that is not normally found in other organisms. So if you define this as a species that cannot cross with another one, they've achieved it without CRISPR. So, and CRISPR will make it easier.
Yeah, and to some extent, phylogenetics, it's a useful way of grouping large numbers of organisms together. But in terms of what that means for us, it can really be down to single genes or a wide variety of genes, but that perform very different functions, even though it's basically the same organism, right? So if you take, for example, pathogenic bacteria, oftentimes they're only going to differ from a non-pathogenic bacteria by a very, very small number of genes. So they are the same species, but they could have acquired a piece of DNA that contains a few genes that causes disease, a horrible disease in humans, while when they lack those genes, they're completely fine in their interaction with humans. So I wouldn't get too hung up on phylogenetics specifically, right? So you could have a quote-unquote different organism as measured by phylogenetics, but we don't necessarily know what the function of that organism is going to be solely from that.
Great question, though.
Thank you.
Really loved it. Over on this side?
Hi, so I'm a plant science major from Cal Poly Pomona, and I have a couple of different questions. So earlier it was mentioned that genetically modified food products haven't been proven to be harmful, and I believe that in fact they've been a major economical advantage in the farming industry. Do you see CRISPR having different effects, particularly harmful effects on food products and the farming industry in general? And then my other question is, I think that this could cause a major ethical uproar as far as human gene modification goes. So is this an idea that will stay purely in the scientific community, or do you think that the general populace will actually have a say on this?
Okay, so this is from a plant scientist from Cal Poly Pomona.
Science major.
A plant science major from Cal Poly Pomona. And she's asking about whether or not, the impact that CRISPR will have in general on plants and other food products, right? And then the other question is in terms of using it in humans. So Carolyn?
Great. I'm glad I get to answer the plant section of this. I think that CRISPR has an enormous promise in terms of providing more rapid resistance to pathogens, and I think it has a great promise in allowing us to more carefully alter the genes so that we can have plants that are drought tolerant, for example, pest resistant, have higher yields. I think there's a lot of potential there. For the human part, I'll let someone else take that one.
So the question was whether the public is going to contribute in the decision or whether it's going to stay in the scientific community. And I think what we wish is that the whole community participates and contributes in these and understands what are the implications of these modifications. The impact that this could have is tremendous, so we will be much better off if all the sections of the community participate. As you can see, just with the diversity of the panelists here, we have important comments to make, and there's much more sitting in front of us with the different perspectives on how we see these. So the preferred answer for me would be that the whole community gets involved.
Okay, and I think we're ready for our last question.
Are CRISPR modifications hereditary?
Oh, great question. Are CRISPR modifications hereditary? A short answer is it depends which cells you're modifying. If you're just to modify your muscle cells, that would not be hereditary. You'd actually have to modify your gonad. You'd actually have to modify the eggs in a woman. And if you manage to do that in vivo, which I think would be really, well, that would be difficult, but yes, just as with in vitro fertilization, you could take out an egg from a woman, you could modify it with CRISPR, and then yes, any change that you made in that egg would be inherited on down to all the descendants forever. Another question would be can you use CRISPR to genetically modify sperm? That's a little trickier because there's billions of sperm, and so it's not going to be, you're going to have to do it repeatedly over and over and over again. But yes, in that sense, any genetic modification that CRISPR makes is no different than a mutation or any other change in the genome. And so then in that case, the question is what type of cell is that modification being made in? But if you were to, let's say we found a way to CRISPR our brains to improve our memory, so as we get older we don't have such memory loss, and we could just inject that in there, fine, but that would not be, well, I guess if you're that old, you're not having any kids anyway, all right? Let's say you're younger and you want the math gene to do the algebra or the calculus, and you do the CRISPR to your brain. That's not going to work its way down to your gonads, and that will not be inherited. But great question. Okay, I'd like to thank the audience and as well as the panel once again. This now ends our science lecture series for 2018.